Siddhi Karmaja Udmatma Virmasaha. Success is achieved through the efforts, so never give up. Good morning, everyone. Myself, Dr. Sakshi Bhardwaj, on behalf of Eltem Technologies, I am delighted to welcome you all in this subsequent session of Women's Day special webinar series on Molecules to Medicine, Women Leading Collaborative Science and Informatics. I appreciate each and every one of you for sparing your valuable time to joining us today. Moving towards to our today's webinar on computational approaches for profiling polypharmacological properties and identifying anti-inflammatory targets in Rungia pectinata. Rungia pectinata basically is a small and soft herb with blue flowers as an undergrowth in forest and roadside and waste areas. There are multiple phytochemicals associated with this plant. This plant exhibits multiple pharmacological activities as well. To know more about it, we have our expert of the day with us, Dr. Sanchita Rajkova, Assistant Professor, Dilbugar University. Dr. Rajkova is an esteemed academic and researcher in the field of bioinformatics, possessing a solid background in computational biology. She completed her BSc in Geology and MSc in Bioinformatics from Dirpugar University. She earned her PhD in Chemical Sciences and postdoctorate in Molecular Dynamics Simulation from Tejpur University. Her expertise includes critical areas such as computer-aided drug designing, immunoinformatics, and molecular dynamics simulations, leading to significant contribution to her discipline with 25 high-impact research publications, 6 books chapters, and 4 patents. In addition to her research achievement, Dr. Rajkua is committed to education and mentorship, guiding PhD students and overseeing numerous students' projects that promote practical learning. She has delivered several invited lectures as conferences and has been recognized with the prestigious Upendranath Goigi Memorial Dibrugarh University Teacher Award in 2024 for her impactful contribution to academia. Through active collaboration on research projects, she fosters teamwork in scientific exploration while inspiring future scientists with her dedication to education and innovation. I welcome you, ma'am. I request all my viewers to feel free to drop their queries into the chat box. We will be taking questions and answer at the end of the session. Without any further ado, I would like to hand over the platform to our expert. Over to you, ma'am.
Good morning, everyone. Thank you for the generous introduction, Shakshi ma'am. It's a pleasure to be here today to dig into this intriguing and rapidly development field, network pharmacology. So today my presentation will focus on integrated strategies of network pharmacology, molecular docking and dynamics, and exploring the phytoconstituents of Rungia parviflora pectinata as well, or for anti-inflammatory target discovery. Now, before we plunge into the specifics of this topic, I would like to provide a brief overview of network pharmacology and its operational mechanisms. The purpose of this talk is to explore what network pharmacology is, its historical context, how it has evolved alongside systems biology, the key concepts that underpin it, and its significance in drug discovery and development. Uh, so, the highlights of today's presentation include the definition of network pharmacology, its historical roots, the evolution of drug discovery, understanding the biological networks, key concepts in network pharmacology, analytical techniques used in this field, and an application example. Now, historically, humans have relied on natural substances for healing. So their ancient text documents the use of plants, minerals for medicinal purposes without a clear understanding of their mechanisms. However, as knowledge progressed, pharmacopoeias have emerged, and these are the authoritative books listing medicinal drugs and their effects based on empirical evidence. And they have laid the actually groundwork for systematic drug classification. And nowadays, in the 20th century, it has marked a significant shift in this whole drug discovery process due to the advancement in molecular biology. And thus, researchers have begun isolating active compounds from natural sources and studying their effects at molecular level. Uh, there are many groundbreaking drugs which have already been discovered serendipitously. That means without a clear understanding of their mechanisms. A very, famous example is the discovery of penicillin by Alexander Fleming, which has revolutionized medicine and paved the way for modern antibiotics. However, now, with the focus on specific molecular targets like receptors and proteins, this approach has led to the development of drugs like aspirin and statins, no, which have had like really profound impact on overall human health. Uh, okay, so no, this just a minute. Okay. So, as our understanding of diseases have evolved, it became evident that many diseases are multifactorial actually, and they involve integrate interactions among genes and proteins and pathways. And this complexity has challenged the traditional approaches to drug discovery. And to address this complexity, this systems biology has emerged as a field, and it has aimed to understand biological systems as a whole rather than in isolation. So they are very compressed. They give us very comprehensive understanding of drug actions. And of course, with the maturation of network science and the tools that are provided to model and analyze these complex interactions within the biological systems, therefore this has now brought us to the heart of our discussion today, that is the network pharmacology. So what exactly is a network pharmacology? It is an interdisciplinary approach that integrates biological networks, systems biology, and computational methods to understand the mechanisms of drug action and identify novel drug targets. It shifts, the whole focus from this individual drug target interactions, which we have been doing for quite some time, to a broader impact of drugs on the whole biological system as a whole. So, and network pharmacology, it seeks to identify not only the specific target but also the interconnectedness of how drugs they affect these networks. So this network pharmacology, they have shifted this whole focus, like I told you, from single drug target interaction to the effect of drugs on biological systems, and maybe aiming we try to identify the interconnected targets, their influence on the networks. Okay. Like traditional pharmacology often examines one drug action on one target, but diseases and complexes, and they do not go for these kind of what to say, a very comprehensive study. Like for example, now aspirin, we know it primarily targets the COX enzyme, no, but it also influences multiple signaling pathways related to inflammation and pain. So this has, the network pharmacology has actually helped us to understand these broader drug implications. Networks can be used for generic data sets. for people connect, like for people how they're connected to people. Okay, but in biology, networks can be used for looking at molecular interactions, like connections between biomolecules, connections between cells, tissue tissue connections are there, protein protein interactions, drug protein interactions are there, food webs, ecology, etc. So it has a very wide variety of applications. These interactions can be protein protein physical interactions, they can be regulatory interactions, such as say a gene A regulate a gene B, or maybe genetic interactions like one's mutations no reaction to another mutation maybe. So they allow this overlay of protein protein functional interactions and gene expression data set to see if any connections are there. So this is one example that I have taken from this reference that I have mentioned here, and it is a medicinal herb, core active ingredient, and then key targets and pathway network, and it is representing a very holistic view of the drug effects. So another example is this bioactive network of Triphala, and you can see that the three botanicals of Triphala are represented as the dark green circles, and how they are connected. So these kind of studies are possible, and they are good. What to say? They are in a lot of demand now. So the 6 degrees of separation. Now here comes the 6 degrees of separation, which is a concept that suggests that every person in the world is connected to every other person through a chain of acquaintances that has on average say no more than 6 intermediaries. So this idea originated from social experiments and the work of various scholars. But how are we connected to certain connection? How to know that? So there is a computer science algorithm which is called as the breadth first search, which is a fundamental graph traversal algorithm in computer science, known as the graph theory, and that is used to find the shortest path between two nodes in an unweighted graph. Oh, okay. Sorry for this. It's written that why is network pharmacology important? The importance of network pharmacology. It is important because it accelerates drug discovery by reducing time and cost in drug development. It also helps in tailored treatments to individual patient profiles, and with the they are now in helps with the precision medicine. They also help in the importance is also felt by from these uncovering complex diseases, wherein they help to unravel multifactorial diseases, and of course identify new therapeutic uses for existing drugs. uh, there are several types of networks, okay, that we can consider, like the gene regulatory network, which depict how genes regulate each other. Then there is protein-protein interaction networks, which show how proteins interact within cellular processes. There is metabolic networks, which illustrate biochemical reactions within metabolic pathways. And of course, we have the drug-target interaction network, which represent how drug targets interact with various biological targets. So in our lab, in right now, we are mostly concentrating on the drug target interaction network. Certain key concepts that we should know about this kind of networks is, first is the drug target interaction network. We will come in details. Next is the biological pathways, and understand how the drugs affect specific pathway. The network analysis, like quantifying network properties to predict drug effects, and of course, multi-omics data integration, where we combine data from the various genomics and proteomics and other more data. Uh, so, drug target networks are critical for understanding how drugs exert their effects at multiple levels, and they allow us to visualize which proteins or genes a drug targets with, and how these interactions influence disease outcomes. And yeah, so these are some of the databases that can be used for this kind of studies. So there are several databases. In fact, we use Swiss Target Prediction, and then we have FarmMapper and some other databases that we use abundantly for this kind of studies. Uh, So, understanding how drugs impact these pathways is fundamental for development of target and effective pharmaceutical interventions. It is very essential. And some of the databases that you can some popular databases for biological pathway are the Kyoto Encyclopedia of Genes and Genomes is there, Reactomes, Panther, Biosic. So all these may be used for pathways, biological pathway studies. And then we have network analysis. So network analysis, it is to analyze biological systems effectively. We construct networks using relevant biological data from various sources, like we get these interactions from sources like string maybe, and then we can analyze this involves integrating information from literature, databases, and say experimental results. Just a brief on network analysis. So we in this process we get the relevant biological data to construct a network, and then once constructed we perform topological analysis to understand the network properties, like say for example node degrees there, where the number of connections of a node has, no high degree nodes may indicate say crucial targets. And then we have centrality measures, where we identify influential nodes that play significant roles in maintaining network integrity. Then we can also use clustering coefficients, which indicate how interconnected nodes are within local clusters. So these kind of matrices can be used to analyze your network. And then we can identify certain hubs or super hub genes in the network itself, which have very high degree of connectivity and are considered crucial for this network integrity. And targeting or perturbing these hubs can have significant impact on network behavior and consequently the drug effects. So this is how we can come up with lay down our groundwork of to perform a network pharmacology. Of course we can then go for functional enrichments and other studies as well. For this we can use the open source platform Cytoscape. Then next we can do is perform gene ontologies, which is a standardized system of terms and concepts used to describe the functions and attributes of genes and their products across different species. And it provides structured and controlled vocabulary to annotate and categorize genes and gene products based on their biological roles in various molecular function categories, say, which describe the uh, specific biochemical activities or function of a gene product. Like, for example, it may define a protein as having kinase activity. So it will indicate that it has a role in phosphorylating other molecules. And then we have biological processes. This category focuses on larger biological processes or events that a gene or gene product is involved in. Again, for instance, it may be described as describe a gene as being part of the cell cycle or immune response. Okay. And then we have cellular component, and this category identifies the subcellular structures or locations where a gene product is active. And it might specify that a protein is located whether in the nucleus or in the mitochondria. So all these things can be studied using the gene ontology. The importance of gene ontologies can be felt as it provides uniform technology for terminology for biologists to describe gene functions, and it reduces the ambiguity and also enhances communication across fields. Not to mention that geoterms are used to annotate genes in biological databases and enabling which enables the efficient searches based on gene functions, and you can then go for data interpretation. Of course, it also facilitates the identification of overexpressed biological processes, molecular functions, and cellular components in gene sets, and it enhances the understanding of experimental data, and it allows for the comparison of gene functions across different species, helping to identify conserved biological processes and evolutionary insights. So there are many online servers and tools available to perform gene ontology. Like we have the Gene Ontology Consortium, David, Panther, Gorilla, Amigo, GC, and there many are there. And even there is one plugin in Cytoscape, which is the Bingo, which is also used for gene ontology analysis. So with this, we have come to the application example of network pharmacology. This work was done by Aleha Jahin. She was my student, and this work was done as part of her MSc dissertation. Um, Now, before going into before beginning the topic, let me provide you an overview of the background and context of this particular work. So, Rungia pectinata is a member of the Acanthaceae family and is an ethnomedicinal herb. And there are numerous studies that have shown its potential anti-inflammatory, analgesic, and antimicrobial activities. It is found in states across India, including Assam, Andhra Pradesh, Karnataka, Kerala, Maharashtra, Orissa, Tamil Nadu, and it contains various phytochemicals and has been traditionally used for ailments like smallpox, inflammations, and fungal infections. This plant is rich in phytosterols, terpenes, tannins, glycosides, flavonoids, and other bioactive compounds which contribute to its medicinal properties. But despite its recognized health benefits and traditional uses as a leafy vegetable, it lacks thorough scientific exploration. As you can see from this graph, we you can see that there is very less number of scientific exploration regarding this plant, and this has been the basis of this study. So while this herb has been documented for its efficacy in reducing inflammation, the specific pathways, biological targets, and metabolic processes through which it operates remain very poorly understood. So that is why, through this study, we tried to bridge this gap by providing a detailed analysis of the herb's bioactive compounds and their interactions with molecular targets. And therefore, we laid down three objectives. The first was to identify principal bioactive compounds in Rungia pectinata and explore their potential molecular targets and construct a protein-protein interaction network. Next, we went for the GEO and the KEG enrichment analysis to understand the biological pathways involved in Rungia's anti-inflammatory effects. And then we performed molecular docking and dynamic simulations to assess the stability and to understand and to assess the stability and their interactions. So here is the methodology part. So for this study, we retrieved the phytochemicals from a study that was conducted by Zheng et al. In 2016, and we choose 38 compounds for this particular study, and then we performed the admit analysis of the bioactive compounds using the PKCSM server. And then we acquired the targets that regulated by the bioactive compounds of Rungia pectinata. Next, we acquired the potential anti-inflammatory targets from DisGenNet and gene cards, and then we tried to find, potential common targets between the regulated bioactive targets regulated by bioactive compounds of perviflora as well as from DisGenNet. And we got a nice this Venn diagram, and then we went ahead with the PPI network construction and analysis and performed the enrichment analysis of the GO and KEG. We have used for GO we have used a plugin Bingo from Cytoscape, and then for the KEG analysis we have used the Cobus server. Then we went ahead with the retrieval of the protein structures from protein data bank and performed docking, and then the dynamical studies. These are the results. So like I already mentioned, we have considered 38 compounds, and of these 38 compounds only 7 exhibited notable admit properties and met the criteria for drug likeness scores. That is why we selected these 7 compounds and went ahead with the next study, that is acquisition of potential perviflora and anti-inflammatory targets. So on the left side, you can see a Sankey graph. So the top 20 hits for each of these 7 compounds were selected based on the highest probability. And overall, we got a total of 140 ingredient related targets were identified from Swiss target prediction database, and the compounds and their corresponding biological protein targets were analyzed using this particular Sankey graph. It's also known as a bipartite network graph. And the set here on the set on the left represents the compounds, while the set on the right represents the protein targets, and the connections between the targets and the compounds are depicted by the gray edges. Then we went ahead to retrieve 728 targets related to inflammation from DisGenNet and gene cards, and the genes linked with the compounds were compared to anti-inflammatory genes. And this Venn diagram was constructed, resulting in a gene interaction chart. And here you can see that 22 shared genes indicated potential anti-inflammatory targets of Rungia pectinata. Uh, okay, so after this, we use these 22 genes to retrieve their networks. For this we utilized the string database and imported in Cytoscape and performed the analysis. The figure on the left provides a comprehensive illustration of the network and highlighting both the core and the non-core targets. And as you can see, we found a total of 129 nodes and 442 edges which were observed in this network. And when the further analysis enrichment was performed, we found that it showed 25 key nodes which were identified based on the node degree. And from this 25, you can see the yellow colored nodes. These were identified as the super hub genes, and that has the highest degree of in the network. Yes. Then we performed the gene ontology analysis using the Bingo enrichment analysis and conducted it on the top 25 gene hubs. hub gene, sorry, and which yielded are almost around 86 biological processes, 5 cellular components, and 81 molecular functions. You can see that the bingo enrichment analysis identified significant enrichment for biological processes such as a response to stimuli and metabolic regulation. Also, it identified cellular components like the caveola and extracellular matrix, and molecular functions such as protein binding, signal transduction among the top 25 genes. Yes. Then we performed the KEG pathway enrichment using the COBUS, and figure on the left, which were classified according to the p-value from the small to large, of which 7 pathways were related to inflammation. Among the inflammation related pathways, we found that the interleukin 1,7 signaling pathway has the highest enrichment degree. And from this pie chart, it was observed that among the associated targets, the SRC, PTGS2, and IL-1B were involved in the highest number of pathways. Thus, we considered these 3 targets and performed our next study. This is the construction of the ingredient target pathway network. And as you can see, this was also done using the Cytoscape software, and a network graph was conducted. And the nodes included 7 active ingredients of Rungia pectinata, 22 potential anti-inflammatory targets, and 36 signal pathways. And here we can see that the compound identification numbers, these 2 numbers, are key nodes due to their high degree, indicating significant anti-inflammatory effects. And the core target, PTGS2, is linked to multiple pathways, including the IL-7 signaling, the arachidonic acid metabolism, and the VEGF signaling. Next, we went ahead with the DFT analysis of the 7 compounds, and I will not go into the details because this DFT I have already covered in another talk. Here I would only say that we have used various parameters for screening, and on the basis of these various parameters, and you can see the gap, the homo-lumo gap is also very the lowest in the LGF and LGG. So both these the CID numbers also you can see are also the ones which we found in our target pathway, the network, and we considered these 2 ligands for our docking purpose. Okay, so after the DFT analysis, we went ahead and retrieved the 3 target proteins from the protein data bank. These proteins had some missing residues, and which so we had remodeled and then superimposed with the downloaded or retrieved structures, and we have used the binding site as the one given in the PDB, and then we have performed molecular docking of LGF and LGG with all the 3 proteins, and you can see the C-Docker energy values and all the docking parameters, and this was done using the Discovery Studios. From the slide, the take-home message is that out of the 7 compounds, the LGF, that is the trans neurolidyl formate, and the LGG, that is the withdrawal, showed the best results in terms of C-Docker energy as well as the number of non-covalent interactions. This slide shows the figures of the docking results, and the figures indicate that PTGS2 exhibit the highest number of ligand interactions. Similarly, the figure also revealed that SRC protein forms the highest number of H bonds, and these interactions are all consistent with the binding patterns observed in the native ligand. Uh, yeah. Then we went ahead with the molecular dynamics simulation, and these are the results of the RMSD. And you can see that the RMSD plots from MD simulation show that the ILB1, PTGS2, and SRC proteins are more stable when bound to the ligands, the LGF and LGG. Okay. These are the RMSF results, and here you can see that the ILB1A has a similar fluctuation of key residues as like the just like the native protein. However, in case of SRC and PTGS, you can see that when the protein is in complex with both the ligands, they are showing less fluctuation as compared to when with the native protein. These are the results of the radius of gyration, and it measures the compactness of proteins, and it shows that binding ligands, the LGF and LGG, increase the stability and compactness of the IL-1B, PTGS2, and SRC protein during the simulations. These are the results of the MMPBSA, which was performed, and you can see that SRC ligand complex have significantly lower energy values. This indicates stronger binding affinities as compared to the IL-1B and the PTGS2 complex. These are this figures are of the hydrogen bonds and other non-bonded interactions, and you can, from this figure it can be seen that there are predominant H hydrogen and hydrophobic interactions in the initial complexes. Also, it indicates that sustained stability for LGF and LGG compounds within the binding cavities. As you can see that there is fluctuation, but then they are maintaining their interactions within the binding cavity. Ah. So with this, we come to the conclusion of this work. So this study elucidates the anti-inflammatory and anti-cancer mechanism of Rungia pectinata, which identify key bioactive compounds and their molecular targets. And using network pharmacology and molecular docking and dynamics, this work highlights that narrowly formate and withdrawal are as lead compounds that stabilize key targets, which include the uh, IL-1B, the PTGS2, and the SRC, thus reaffirming the herb's traditional use and its potentially novel therapeutic strategies for inflammation and cancer. And this finding also underscores that this particular plant has potential as a natural therapeutic agent for inflammation and cancer, and it warrants further clinical validation and exploration. So this work recently came out in Journal of Cellular and Molecular Medicine, and yeah, these are some of the references that I have used while preparing these slide. These are the students from the batch where Aleha was, and you can see her in this yellow mikla chadar. There are three yellow mikla chadars. Okay, I am wearing yellow, and then she is the yellow on the right is Pinky Francina Lafalo. She is from Lesotho, and she was a foreign student. And the other yellow on the left is Aleha, and you can also see it on the picture below. She's on the extreme right. Ah, thank you for listening. I hope I have finished it on a record time. Thank you so much.
Yeah, thank you so much, ma'am. It was very interesting session. So let's take some questions now.
Yes.
Okay, why network pharmacology is important from drug designing?
Yeah, like I told you that when we say drug design, we normally feel that maybe one protein interacting to one drug, right? Or a single drug can have only inhibit, can be used for a single disease. Say. But with the help of network pharmacology, now you can think of broader. You can explore the broader sense. You can find out if it has multiple uses or can be repurposed, and so that is why it is very important.
Correct, correct. Yes. Next question. How do you ensure the accuracy of compound target interaction derived from these databases?
Yes, these yeah. So when we derive these these data from databases, so they give us certain p-values, and we use that. They have certain scores on them, and we use it. And of course, we try different databases and always, come to a consensus as to what it can be.
Okay. Next question. If two or more pathways, how to decide the target?
Yeah, it can be. Like in case of aspirin, I told you, so single drug can go for multiple targets. That is the basis of network pharmacology.
Okay. Polypharmacology suggests that a single compound can interact with multiple targets. How do you assess the potential? I think this is the same question. How do you
Mhm.
assess the potential of a compound to exhibit polypharmacological properties, especially in the context of anti-inflammatory activity?
Yes. So in this study, actually, I told you that in pharmacopoeias it has already been reported that Rungia purviflora or pectinata, it is the synonym, they are used for anti-inflammatory activity. But they were not, a more scientific approach was not done, or which exactly is the target. Those kind of studies were also not done. So, yes, with the help of network pharmacology, we can come to it. And of course, nothing can be proved without experimental evidence. That is one drawback that we have in our lab. But of course, with collaborations, maybe we will try to cover this part of the study as well.
Okay. Next question. Okay, so I think we have done with the questions and answers. Thank you so much, ma'am, once again for answering all these questions and for the great presentation. It's
Thank you
time
so much.
for certificate of appreciation and memento for our guest of honor. Yes, we
Thank
are
you so much.
grateful that you could have joined us today. It was a pleasure to have you, all my viewers, as well. To stay tuned with us, like, subscribe, and follow us. Thank you so much. Let's connect and grow together. Thank you.
Thank you so much.